It started. Hi everyone, I'm Yi. I'm an associate professor at University School of Madison. I'm the moderator of this panel session, Bring Connectome. First, I want to thank Jie and Hongtu for organizing this wonderful workshop, and also thank you all for coming and then staying to the last panel, last session of the last day. First, I want to apologize that there are some changes to the panelists. So today we will have Dr. Chen from University of Maryland School of Medicine, Dr. Chung from University of Wisconsin Madison, and Dr. Ivor Cribben from University of Alberta, and then Dr. Chung from University of Pittsburgh to give us their insights about brain connectome. Just to give, I think after 4 days of workshop, I don't need to say too much about brain connectome, but still I want to just provide some quotes from Sporn's 2005 paper. They introduced the concept of human connectome, which is a comprehensive structural description of the network of elements and the connections forming the human brain. It is fundamentally important in cognitive neuroscience and neuropsychology. And then the connectome will significantly increase our understanding of how functional brain states emerge from their underlying structural substrate, and then will provide new mechanistic insights into how brain function is effective if the structural substrate is disrupted. In general, when we talk about brain connectome, so based on the spatial resolution, so we may consider 3 different scales: a macro, meso, and a micro scale. I think we have an excellent talk about the macro scale from today's keynote. I think most of us are focusing on the macro scale and using some non-invasive imaging techniques such as diffusion and functional MRI to study human brain connectome. So today we will have some questions focusing on more on the scale, and then see and then get some insights from the speakers. So the first question I have is kind of what we are basically it's does what we measure or quantify, for example, what we usually see the functional structural connectivity map real brain connectome. So basically, I think my answer is no, but still, so because we use those measures to do the study, and then I think the first question we want to understand is what we are actually measuring, and then also what we are measuring, is what we are measuring have some like a clinical meaningful, and then can we translate into some like a clinical impact, and then also, for example, from a pathological point of view, suppose we have a marker, and then we want to have some like a treatment on the targeting the marker, and then what kind of brain connectome or brain connectivity we are targeting on. So I hope the panelists can give some insights on this. Can we start with Chung?
There you go. I'm not sure what
you should have told him he had to speak.
I mean, brain imaging is not the direct measurement. It's basically summary measurement. So that's all I can say. Yeah.
But you have done a lot of like imaging analysis so far.
Yes.
And you're trying to extract some features, right? Like topological features and all the things. And also you're trying to link all these features with like a clinical diagnosis. You're trying to distinguish them.
Yeah,
Do you believe that all these analysis you are doing are meaningful for clinical purposes? You need to answer questions, okay?
When I was junior, I didn't believe that. Actually, I just did basically math stuff. But now I believe it. That what I'm doing is actually very meaningful to the actual clinical research later. Possibly not directly related to clinical research, but for example, in fMRI, resting fMRI, the clinical relevance of resting state fMRI is not much well established. I mean, there are some bunch of papers, but if you measure something on a resting state, that doesn't directly translate into the clinical development, research development. So, but I think there's some connection there. It's just that someone have to sit down and do establish some connection, I guess.
No, I think you think about this way. As in the current literatures, most people just focus on calculated correlations or dynamic correlations. And also try to extract some summary statistics from this correlation matrices. But why do you feel that your topological metric is more meaningful in what sense?
Oh, that's the technical question I can answer definitely. The correlation is, if you look at it, the existing literature, basically they use pacillation, correlation of the pacillation. Before you that, you do the averaging of the time series in a pacillation. That's the central limit theorem. So you kind of correlate things. So kind of then you are correlating. Because of that, you are essentially artificially inflating the correlation. So a lot of signal you detect in the literature is not it's not actually real signal, it's mostly false positive actually. That's my understanding. So in order to fix that problem, your connectivity measure has to be additive function. Correlation is not additive function, because additive function over the set. So the one way to fix that problem is your connectivity matrix has to be something like entropy. Entropy subtracted by something, some common set, such that your function connectivity is additive over the pacillation, then your correlation measure will be more meaningful. Your connectivity measurement more meaningful. That because of that, if you look at the structural connectivity, you're basically counting the number of fibers. It's addition, so it's additive function already. So structural connectivity based on the pacillation usually has some meaning. It's actually more reproducible, but because of that, if your connectivity matrix is, your connectivity is basically correlation, most likely not gonna be reproducible. It just implant the artificial signal there. So so basically, you should not use a correlation to do the resistor fMRI. That's my understanding.
My point is, how would you validate your statement? You can say whatever you like to, right? You need to use something like either real experiment or like I have the.
Oh, I can show you that but if you do correlate the topological based approaches, you can actually reduce the variability significantly, and compared to the traditional correlation based approaches. What I'm saying is that correlation-based approach is not the way to go for the rest of them, right? So because of that, the Honendomba is actually trying to use entropy, transfer entropy, I think. And
why do we need to trust him?
He's not here, but
why should I trust him? Because you can claim yours, I can claim mine, right?
Oh, I'm actually writing a paper demonstrating the correlation-based approach will actually essentially inflate the signal. Okay. And I think most likely that's a correct statement. So all the literature based on correlation on the pacillation might be inflating signal dramatically. One way to avoid that issue is that you have to do the correlation-based approaches at the voxel level. Then maybe okay. But if you're using pacillation, the pacillation itself is a biased model. It will give you biased outputs.
I'm not quite agree with you, because the pacillation, you think about is a kind of global kinds of the modeling of the breadth data. It's a kind of first level of how to summarize the information, right? And then if you go to like the contracted, it due to the neurons, you're too small, okay? You see all the tiny things, you don't get a global pictures. I think that to me is most of neuroimaging based on template and the atlas is kinds of more global kinds of the modeling, the complex data set that we are working. You see the point? That's the reason Atlas is still one of the most important research topic in neuroimaging data analysis.
That's what I'm saying. That framework is wrong. That's what I'm saying. that the parcellation-based framework is totally wrong. That's what I'm saying.
See, okay. But that's important, right? That's the discussion is important. Okay, let's go to next.
Thought it was just going to be discussion between Hung 2 and Mu, and I was happy with that. What are we measuring? I don't think it's an indirect measure. If I mainly work on fMRI, so it's an indirect and incomplete measure. The real brain connectome would be looking at the neurons and synapses and looking at the connections between them, the chemical, electrical, long range, short range connections between them. What frustrates me is that, you know, we have a GLM for looking at brain activity. Even though there's a lot of problems, as John pointed out, what we don't have is any sort of GLM or standard method for estimating connectivity. So in my case, functional connectivity. We don't have any standard. We started off with correlations, simple Pearson correlations. We dabbled in sparsity, and now I feel as though we're back. Most of the papers that I read are back to correlation again. Mu doesn't like correlation clearly. And then Wes talked about that in terms of prediction, correlation was doing a better job compared to sparsity, but in terms of interpretation, sparsity, a sparse correlation matrix or partial correlation matrix is easier to interpret. So it would be great if we could agree on some sort of standard functional connectivity. I'm not going to talk about structural connectivity, but functional connectivity pipeline. I see a lot of papers where they don't even whiten their time series before taking their correlation and doing testing or doing inference on the correlations. They'll just estimate the correlations and then do testing straight away. So I believe we should whiten the data. There's, you know, what sort of pipeline can we agree on? In terms of combining the data, I would like to see a little bit more what different people have talked about, combining the structural and functional together, one jointly, either jointly or one inferring the other. That would be, I think that was something that I think we should concentrate going forward. I think what Shuo does is great. I'm going to shut up now in a minute, but in terms of clinically meaningful, so we get a correlation and an association. That's great. You know, between say disorder and what do we do with that? I mean, I'm a part of this. We all have these great methods for estimating dynamic functional connectivity. We disagree on the best methods. Well, let's say we did agree on the best methods and we have this correlation. How do we bring that to the lab? What do we do with this information? Is it stimulation? Is it what Rob Cass was talking about, like nanobots, you know, reconnecting? What do we do with this information? In terms of connecting to the clinic, I see two options. I see we either go into the clinic to communicate with those people, or we train the clinicians to, you know, use this information that we generate. And they're the two options that I see. You know, we either educate them in what we do and how we can help them, or we go into the clinic and start communicating, being that bridge between our methods and clinical usefulness. I'll stop here.
Thank you, Ivor. And trying to answer the first question, I think due to the limitation of the current measuring technology, we are not so sure what really measuring. For example, the functional connectivity, as mentioned, mainly is the correlation between the time series. And the time series could be the both signal, and even the both signal, I think Gang just discussed, what the measures could be a mixture of the neural activity and the physiological signal, like your heartbeat and others. And but from other perspective is the whole neuroimaging community, we see that based on this postulation and the correlation method, we see a lot of reproducible results from the starting from default mode network, then to the YAW, so 2011 and 7 and 17 network, we always see these results from this study and from that study. We always see that this is give us some confidence to say there are some signal can be replicated across studies. And the similar thing, I think, from the yesterday or Wednesday's talk, I think is from the white matter tractography, we can recover the most important fibers from the tractographic analysis. But always there could be a lot of noise there. And we're not sure exactly where marrying is covariation of the neural activity. Can we say that? I don't think so. Yeah, but could be some signal combined with the noise there. So that's yeah, I don't know how to prove that, but it's just my belief. And yeah, we just recently we did some analysis using some methods to see whether the findings from UK Biobank can be replicated by the HCP aging study. The findings are highly replicable in using the correlation postulation related method. And I think it depends on also what your methods use, what the data quality is. But because both HCP and UK Biobank data quality are pretty good. So if you change to another data set, you may not see the high replicability. And the last I want to talk about is the clinical applications. We happen to have 2 clinical applications with LH in our group. One is both of them are using TMS. So they found something in previous findings that kind of the smoking behavior in schizophrenia is about 60% of the schizophrenia patients they smoke, which is almost 3 or 4 times than the common population, general population. And they try to identify that brain areas have a abnormal connectivity that is related to the nicotine addiction. And then they focus on that, design some coil to do the TMS, and then run a clinical trial on that. And the other similar trial is on hallucination for patients with schizophrenia. And the first one, we finished most of the study. We do see some reduction in the functional connectivity. So there's some alteration of that as a kind of immediate part of the smoking behavior change. It's only 60 patients. It's just just a pilot trial proof of principle. But what is really going on there, I don't know. But it's just some good pilot study to see that. So that's my experience with the clinical application. Yeah, that's it.
So first, I want to mention that I'm here just to fill in the missing person. So imputation. So
You do.
So just share some thoughts. So I basically share the similar idea. So it depends on what the data you use. So if you say that you use a functional MRI or MRI for measure the functional connectivity, the short answer is of course no. So I think the true functional connectivity about the underlying mechanism have to be based on the data that measures the neuron firing. So at least you need to get the data from local field potential. So that we do see that if you have local field potential data, you can better actually see the neuron firing directions. So, and in fact, almost all the neuroscientists I've talked to, they say they hate fMRI. And the reason is that they don't think fMRI can give them any basic understanding of the mechanism of the brain. So the short answer for understanding the connectivity of the basic mechanism, fMRI cannot do that. But however, I think they still measure something unique to the person. So you it's very, even though it may not come, many of those measurements maybe come from the cardiovascular activity of the persons, but it's still unique. So it's possible that that unique information can be used in the clinical trials. But the problem here is, I think this is something both Yi and Shuo we talked about yesterday, is like, remember collecting fMRI, you need a lot of conditions. And within that, short of just within 1 hour, can FMI capture the signals that the subject is indeed during the disease status? So for example, if the subjects are in depression, or they have this, or they have some brain disorders, but if the subject are inside the scanner, they're actually in a pretty stable state. So is that just like 15 or half-hour scanning really capture those brain activities when the subjects are under this abnormal states? Maybe not. So it's likely that it may not be the FMI, even though it contains a lot of noise, but it also depends on the scanning status and whether the people who scan, and also a lot of scanning parameters, that's why pre-processing are very important, and whether the person who did the scanning implement a lot of protocols correctly. So I once watched people who did the scanning. I think a lot of other factors can influence the results. So it's not a clean data. And going back to Professor Moon's comments about the parcellation, yes, I agree, it's wrong. But all statistical models are also wrong, but they can be useful. So I think brain parcellation is a very effective way for dimension reduction. We have to do some dimension reduction in order for us to perform computational efficient data analysis. Okay, that's just my 2 cents. Thank you.
Can I respond? Because actually, comment Ivor had. It's something that I've talked with people a bunch about filtering, whitening, right? And I think actually you don't want to whiten, because whitening we do in the general linear model when you have a task, and then generally squares says that the optimal way to detect y equals x beta, beta coefficient is to whiten. But in resting state, I mean, the noise is the signal, right? We don't know what is, and so if you whiten, what you're going to do is you're going to down emphasize the low frequencies and up emphasize the high frequencies to get a white spectrum. But we know that most of the bold signal is down in the low frequencies. So I think if you whiten, you're just going to get a less efficient estimate of the functional correlation, functional connectivity. I don't know. What do you think about that?
And then what about for inference? Just correct for it.
Well, for inference, you should use my XGF method.
I know. I was going to say that's what I was going to say. You're going to use the longitudinal model that you suggested.
Yeah. No. So I think that is the right thing, because you don't know what the since we don't have a model, the noise is the signal. Yeah, just use correlation and for inference. Adjust for the temporal autocorrelation in the one node, temporal correlation in the other node, and the cross correlation. That's what the F-Uni and Nichols XGF method is. Which actually, where's Mark Ficus?
He's not.
He beat me to public. I'm so angry. He beat us to the publication with that idea. Well,
yeah, but I was on that too. I think you're more cited than we are.
That's the point.
Yeah, it's the Roy correction. Yeah.
Yeah.
No. It's a paper from the 70s, I believe. Yeah.
So I work a lot on brain connectomes. I also want to make a comment. I want to use that knowledge. If we want to observe the universe, right, we can use our Bay ICEs, we can use telescope, we can use other devices. With different devices, you see different results. Putting them together, we can understand it more. It's the same for the brain. Use functional, structural, other MEG, EEG, and so on. You all got different signals, but it's the same object we try to observe. And synthesizing all those together can enhance our understanding. I see a trend that, myself, I work on structure a lot, but I start to integrate other things into my pipeline. And many people also just focus on one image modality. What I encourage is we should look at everything together, and so that we can understand more. So that's my comments. So to the first question, what are we measuring? Each one is measuring differences, a mixture of many different noises and signals together. So we don't know how to disentangle all those signals and noises in them. But the sense we're measuring are clinical meaningful, because we see that they can predict behaviors, and they're related to other measures like genetics and so on. So put everything together, we should have a good picture on what's going on, and that's clinically meaningful.
Can I just, I don't know what we mean exactly by clinically meaningful. In my area, when someone says that it's clinically meaningful or useful, it means that there's a chance that insurance company are going to pay for the analysis. And are we there?
No. I think also maybe it's low, but I think that's the people are working on toward that, right?
Yeah, I'll just say a little bit more about what I know. That there was an initial excitement about functional connectivity from clinicians, and that's gone. So it goes to the back of the pipeline for any MRI stuff. Ting brought up clinical trials. Until you know what you're measuring, there's no way the FDA or anyone's going to accept it as any kind of biomarker.
I don't work in functional connectivity, so it's going to be a naive, ignorant question. So if it is, just tell me, I'll leave. But Shuo brought up something about heart rate. So I'm wondering, Tom said that we're interested in low frequency signal, but fMRI or this resting state fMRI is sampled at a very slow rate. So is there any aliasing issues here? You know, you have to, the Nyquist frequency is twice the, you have to sample at twice the frequency of what you want to detect. So low frequencies, we have low sampling rates. There could be aliasing going on here, I think. But I've never heard this whole thing. I've never heard anything about doing any anti-aliasing filtering or worrying about the Shannon sampling rate or any of these engineering signal processing ideas.
Yeah, maybe, but eventually if people do sliding window, which is basically averaging operation. Sliding window usually most people use 40 seconds. We can do it like say 20 seconds to 40 seconds. So that's gigantic sliding window. So I think whatever aliasing effect we have, it maybe wash it away, I guess.
The people who do physiological noise correction are aware of the fact that we generally are not well sampled. And so they know that we have a very challenging job doing physiological noise correction, and they try to do everything, various methods to try to correct for it. But
So are you saying, Tom, before we get the data?
yeah, so for example, the Human Connectome project data, they, in addition to, I think there's some physio corrections, but they also do an ICA-based cleanup. So it's basically, we know it's a mess. And so they use some techniques, including ICA, to try to find those physiological signals, which if we were sampling at, 10 times a second, they'd be nice, well isolated peaks, and we could really identify them, maybe even use very simplistic methods to clean them up. But instead we use ICA to try to find them, or other things, like some people take averages inside the ventricles and other things to try to get what you think is pure noise estimates to regress these things. But yeah, we know they're not being well sampled, and we try to do the best we can.
So this goes to Han Tu's point and your point that maybe we don't do pre-processing of the data ourselves, but we should know what's going into the data that we get. Want me to repeat that? I agree, because you need to understand your data. Otherwise, you don't understand, you don't know what you're doing. Okay, we can move to the second one.
The second question, I think we mentioned this across the workshop multiple times, like it's about the reproducibility and the reliability of our findings. I think maybe the panelists can discuss both on the brain connectome itself and then also some connections or relations with other outcomes that's like clinical outcomes.
All I can say is the problem is with the pacillation. Because we are using pacillation, that's why you cannot reproduce. Everybody basically using different pacillation, basically. So that will be also the problem. And I don't think I actually explained this. Think about it this way: if you have a bar model, AMSIS model, you combine them, bunch of the region, like 10 regions, average them, 10 region average them, 10 region average them, compute the correlation. What will happen is that maximum correlation will increase. You can show it mathematically. That's correct statement. So if you're doing using pacillation fMRI, you essentially artificially implementing signal. There's no signal there. So my estimate based on simulation is that about 30% of the signal you observe using pacillation is basically false positives. That's about 30%. So what the literature you see read is most likely false positive. That's my understanding. So all the fMRI, the connectivity study, especially recent fMRI, in the literature, most likely those are result on the false positives, basically. So if you want to use a pacillation, you should not use a correlation. If you want to use a correlation, you should go down to the voxel level. That's my take. Now, if you want to use a pacillation, you have to use a connectivity function, bivariate function that's additive or modular. Then you will not have this kind of the artificial inflation. So yeah, that's. I'm actually writing a paper on this. So you will see that this paper probably a year from now, I guess.
Okay. Who will read it? I ask the other members to comment on that.
So
I'm not going to invite you again.
No, next time I'll ask him to give a talk, not give as a panel members.
The pacillation section, but others.
After there's a free sections.
So the way I interpreted this question as we should define what we mean by reproducibility and replicability. So to me, reproducibility is the ability to take somebody's code and data and to reproduce the results that they obtained. And replicability is, I think, what Mir was referring to. If you get a new data set, you use the same parcellation, you get the same results. I'm going to comment on the former. So we looked at, in the study, we looked at what I would call computational reproducibility. So looking at taking the data and the code from people's studies in stats journals and trying to reproduce the results. And my student looked at this and they found that they identified 93 papers in the top stats journals. And they could only reproduce 15 or around 15% of those papers.
We need to record that.
You need a record of it?
No, we need to record it and send it to all the editors.
Being recorded.
It is being recorded. So this is a problem, clearly. I presented this at Oaxaca last year and we were all surprised, and neuroimaging data, there's certain things that you can't share, but a student of mine recently did it on business and economics, a more recent study of reproducibility for business and economics in stats journals. So just the application to business and economics data published in stats journals. And we're okay. It's roughly the same percentage for those where the data might not be protected in some way. So we need to do a better job at giving our code and our data to the journals. Obviously, this has to be, in my opinion, this has to be at the journal level. If we're not going to do it unless the journal says it. But there are advantages to doing it. You'll get more citations. People will compare your method to theirs, et cetera, et cetera. So to me, it has to happen at the journal level. If the journals say that you need to have clean code and clean data, you will do it in order to get published in the top stats journals. So that to me is the sort of computational reproducibility element of this. This aligns to what we've been discussing before about when you're sharing your data, giving the raw data and the pre-processed data, and ideally the code to get from one to another that is clean and easy to use. Obviously, that would be ideal, not just one or the other. And this also relates to, and this is a topic that we discussed during the week of, we don't have any benchmarks. We've got no benchmark data sets that we can compare our methods to. And, there's pros and cons, there's no ground truth, but at least it would be a start. It would be a start. John said simulations, we should not do this. I agree. It's always going to favor your method. But if we have some benchmark pre-processed data sets that we could compare methods to, it's a start. It's not the end. It's a good start. So in terms of replicability, in my experience, it hasn't been that great in terms of looking at ICCs across test retest data sets. I think Fleiss in 1966 said we need an ICC of about 0.7. In my experience, we're not getting close to that in terms of fMRI functional connectivity, just to be specific. I'll stop here.
Yeah, I want to respond to some of your comments regarding to the computational reproducibility. I think among all the stat journals, maybe JASA is the only journals do the reproducibility. All these top suggested journals and also no second and tier suggested journals, nobody do it. The reason is there's several aspects. One is, and the journal does not have kinds of the resources to hire people to do it. That's number one, because you know the all journals it doesn't really make a lot of money except the top ones, and the society doesn't want like ASA don't want to give a penny to do that. That's the reason we create AE for reproducibilities. Even this, the title called AE for reproducibilities, they fight it for a while because they say that AE only reserved for the people review papers. They have the prestigious high standards. They are not willing to give the AE for reproducibility. Okay, that's the. But I think that eventually the JASA is willing to do it, and actually the JASA only do it on the ACS side for the first several years. Then the JASA theory method say we're going to follow that, okay? But actually, if you go to the natural journals, sister journals like Natural Methods and Natural Genetics, all these top natural journals, they have similar standards, okay? They have the specific requirement regarding to the reproducibility. You have all these regarding the code, other things. That's in generally, there's the current golden standard in biomedical science, okay? The natural science all have the similar kinds of requirement, or even same. Okay, that's important for whole communities. If you say I do whatever I want, okay, and say I could achieve higher performance in terms of what? Okay, I think this is very important. And also this give the resistance from both statistics society. One of the key reasons is if you really emphasize reproducibility, they have the high requirement on the coding side, computational side. And either because many our students prefer writing papers and without really focus on coding, and then another trend to do that. And this is the problems with our society, okay? In the last 40 years, many of our students come from background from mathematics, and they are not trained to do high kinds of quality coding. And some people resist them to do that, okay? And that's the issues we need to solve. And maybe. And regarding to the second part about the data reproducibilities, I think that's the part is more difficult. And because the first one hosting the large data set is require tons of resources. In particular, now the data set most of we analyze is huge. At least it's not petabytes, so at least on this days all the data we have terabytes data set. How could we have such resources to host them like Amazon cloud, and also how to pay for that? That's the most straightforward things. The second thing is because there's a lot of restriction in terms of data sharing, individual level data sharing. You can share summary statistics, okay? But if you share the individual data set, regardless your process or not raw data, yeah, that's illegal, okay? You break the rules, okay? That's the reason you generally, and also, even though you can create essential resources to other people to upload the data like the cloud, because let's say 5 or 10 years, I believe that most of the computing will done at the cloud, okay? Under the cloud platforms,
I don't feel that you really need to download the data set. We need to come up with rules to say that regulate your just give me the link of your data in the cloud. And then you can other people can go there to test the method on that data without it downloading locally. I think that's the will be the eventually the solution to that if everything goes to cloud. Okay.
I don't fully agree. I think this feels a little bit like over-regulation to me. And like we're in the business of science, and sometimes a paper can be, I have a really great idea and the code might just be a little bit loosey-goosey, but it's still a really important paper and should be published. So I don't like this demand of like anybody who publishes a paper has to check all of these boxes exactly.
That's okay, but that's the different opinion on it.
I think the checking the reproducibility of at least for the data analysis, it's pretty important. Yeah.
I'm not arguing that. I'm just saying, don't make the box too small.
And so I think in terms of noise, we always want to separate what's the true biological variance with the technological or technical variance, which can be just the noise. So I think Ivor mentioned SEC or other methods we can testing. We don't know what really measuring, but what we want at least is if the same subject or same stuff we measure several times by different techniques, at least the signal should be reliable. So that's kind of testing, retesting, or SEC. We want that to be reliable, including that could be a guide to choose pre-processing steps. For example, whether you choose different type of just steps to do the pre-processing to make it more reproducible. And last is the replicability, just we have different data sets, whether we can have similar results. I think I have a couple of slides to show that. It's just a share of the recent, just we can go to the, we can go a little bit to the, for that is we did some UK Biobank and HCP aging study and try to see which edges of the functional connectivity using certain postulation is can be reproduced. And so we did some L0 shrinkage-based subnetwork extraction. We tried to extract subnetworks related to age, and the results are highly replicable. I think most of the networks, there are 2 networks for each of the data set, and then it's pretty similar to each other. And then even we map to YAO's network, the finer postulation, the finer annotation even have a very similar structure. I think it's structured between the sensory motor and the ventral dorsal and others. The structure is highly replicable. And if we only do the kind of individual edge-based analysis, if you go to previous slide. So if we choose the most significant edges in UKB or HCP, the overlap will be just very less. So if we add some constraint to the findings, rather than connections in individual edges, if we do kind of object-oriented kind of findings, the results are more replicable. And this is just one atlas. If we go to another atlas later, we use another atlas. The results are still very similar. So we use Schiffer's atlas. But we don't see the second small network because the first network have a subcortical and the basal ganglia stuff is related. The second one, we don't have that. So most of the network, again, is highly similar to each other, the network related to age. So I believe there is some replicability of the connectivity analysis. So just
I'm happy to see source results. So I think generally I feel so if the pre-processing have been done correctly, I think at the population mean level, the results are reproducible. So for example, default mode detection are very simple example. So you can see this at the population mean level. However, the problem lies in the subject level. And I think FMI or even diffusion MI is a highly noisy. So this is related to John's answer. Why it's not a clinical applicable is because the signal in FMI at the subject level is so low. So that's why I think at the population mean level, if you have sufficient data, so the recent publication said the brain-wide association studies with behavior, you need thousands of studies in order to have a replicable results. And I find that not only you need so much data, also the association practically is very low. So that means that, okay, so for science study, we can have reproducible results, but for clinical application, we are still far from there. Thank you.
But I think I also to Chinese questions, because I think the structure connectivity is pretty reproducible for the same subject. But the functional, of course, is much lower for that, because you can look at the UKB, the 2 time points data sets. You can calculate all the measurements.
That's because you guys are using correlation. That's why it's very low.
We can try your method afterwards, okay? But don't brag yourself, okay? That's easy for us to do it, okay? I can write a paper with him and just issue whether his measurement metric is really reproducible in one sense. Because these days, because many data set has these 2 time points measurements, and also we can validate the things across multiple large data sets. I think this is a great opportunity when really to investigate reproducibilities, all the important measurements and the biomarkers, and through the different studies, integrate information together. I think that's the great opportunity for us these days. Okay.
Can I ask, is structural connectome really that reliable? Because I thought there was like so many different methods of building the tracks and stuff that I thought it's still up in the air, no?
No, I think they already Max, they organize a lot of activity events basically to validate that. I think the kind of things is major track is very reliable. Like a function, the TPSS, the readability is almost 0.9 something like that. I can show you all these. I have the results because there's when you fiber track close to gram meters, of course, reproduction become less, and as the tracking become much difficult. That's the reason the Max developed the method. You see, the Max did when the fiber track close to the cortical surface, and actually you have to additional information, other things to make sure the tracking is okay. But that's repeated is very high in general for the major fiber bundles. Yeah.
But it's very low for the smaller ones.
Oh, of course, because and also when you have these crossing fibers, and of course they but that's cover a lot. No, you don't need a perfect, right? And the technology is not good enough for that.
Could I just comment on your JASA comment? So yeah, JASA introduced the A for reproducibility in 2015. I'll refer you to the paper to see the results from 2015 onwards. The results are at best mixed, I would say. The second comment, of course, there's going to be limitations to this big data sets. Of course, how are people going to download it? Where is it going to be stored? All these problems. Who's going to check it? How are we going to check it? I mean, if people are serious about this, there's different ways of doing it. Granting agencies, matching with some top biostat departments. To get PhD students to do this. There's possible ways of doing this, but yeah, in terms of accessibility, code is obviously accessible, but yeah, where to store the data is a real problem, especially with the data size that we use. I agree with John. If the idea is really good, perhaps it should still be published, but perhaps there should be a star beside it saying we cannot reproduce these results. So take them with caution.
I can make a comment on well, not necessarily on what Ivor just said, but more in general with reproducibility. I think we also have to define what do we mean by that, because reproducibility of the methods versus biological reproducibility. And what Hong Tu just mentioned with TBSS in the structure, yes, we can reproduce it maybe at the voxel level, but when it comes to the tracks, when it comes to the actual measures, and especially if we go into higher order, not DTI analysis of diffusion weighted imaging, but higher kind of more sophisticated methods, that's not necessarily reproducible at the level, at least that we would like it to be reproducible at.
Do you mean replicable?
Well, yes, that's a good point. So for me, reproducibility is like, you know, if there's a biological process, if it's actually reproducible in another experiment, it's not necessarily replicable on the same data set. This is the games we are playing, yeah, with numbers. But I care about science, so you know, I think this next level would be needed.
I think regarding to your comments about JASA reproducibility, actually you can read the recent there's a paper about the perspective about the JASA effort, the effort of basically introducing the AE of reproducibility. I think the published last issue of JASA, something like that. And actually, I think this group of AEs is great. And also they have been spending tons of hours on basically produce that checklist. Okay. And it's not perfect. But I think that checklist was took over and to transfer to other top journals. That's what is I think that effort is super important. We cannot just in general as a field, and you're really doing applications. You want the methods, and all these you need to have high standard for everything you're doing. Otherwise, you'll lose the reputation eventually. I think that's the case. But other things that we can, because so far NIH and all the funding agencies haven't considered that the issues, basically in terms of sharing the data with the public, in terms of doing the reproducibilities. I think at the end of the day, maybe in 5 or 10 years, I think NIH should have a joint effort to basically put the data on the cloud and allow the people to test all the methods.
I just realized the way to make reproducible with correlation. Correlation is not in the space of the Euclidean space. Right? So you have to use the log Euclidean distance to make.
I propose that, but it's better if you do it.
So people mainly use add correlations when the correlation is not should be simply added. So you have to add it in this manifold setting. I think most likely we have to use a log Euclidean distance to add the correlation and do the old operation. So there's some divergence from the Euclidean space to the manifold setting. I think that might be the reason for all those unreproducibility with the correlation.
Your mind is different because I think I don't argue with you on that aspect. Let's see what's going on outwards. Okay. Anything we just conclude? Oh, yeah.
A quick third question about brain connectome analytics. So I put it this way: statistical approaches versus AI and machine learning approaches. Even though many may think they should not be in a competing role, but based on my own experience, when I talk to collaborators, so seems to a binary choice. So either do a regression or some machine learning technique. I'm just wondering if panelists can comment on some like a current practice and then future directions about this topic.
I think you need to be specific in terms of what kind of downstream analysis your people are trying to do.
Right. So I think when I talk to them, so I think most of the scientists that I work with, so they are more interested like in mechanism. So most of the time we still go more traditional, like trying to understand what's going on.
Comparisons or correlated with some.
Yeah, and also correlated with some like a clinical outcomes. And then, but if we go to prediction, that will be another story.
Oh,
You use the topology method.
oh. So what was the question? Sorry.
That's a riddle there. Statistical or against the AI.
Topologically.
Oh, you're missing a one major approach via topological data analysis. And we can at least show that the variability, if you do the average operation in the correlation, variability, if you have 500 samples, variability is reduced by 500 times if you use a topological averaging operation compared to the just averaging correlations.
We'll see. We'll do comparison later.
Correlation is not the king of all the connectivity. That's what I'm trying to keep saying. There are a lot of different ways to get the maximum output. So we either have to change the correlation measures or do something like a topological approaches to basically have the improved. Then there will be more clinical relevance because we can actually inflate the signal dramatically with topology actually.
I have a question for you, Mu. Is there anywhere where TDA doesn't work well?
There's a situation where TDA doesn't work well. It does amazing job when the traditional method actually fail, but it will fail dramatically in a very simple task where traditional method like regression do work very well. So it's not the methodology that works for every situation. It only works for a situation where you cannot solve the problem. If you see an impossible problem, it will solve it. For example, in a resting fMRI, you cannot align it. There's no synchronizing point, right? So you cannot simply average a resting fMRI. With the topology, I can match my resting fMRI to your resting fMRI. We can do that with topology and get a signal and make a meaning. That's one of the impossible tasks with any kind of methodology, known methodology. So.
Okay, to answer this question quickly.
I'm leaving. I just want to say something. Whatever you do, it's going to be statistics at the end.
It's about it.
I believe if we're looking for interpretation and meaning, statistics, prediction, machine learning, and I think where we can come in quite useful is, adding our statistical knowledge to these machine learning methods. Stop here.
Yeah, I think I agree. If we are looking at the mechanisms and the population level inference, statistical methods, and if we are interested in individual level prediction, machine learning methods, but for my research, I think 90% are still statistical methods. Yeah.
So I think Hongtu and also Professor Chen may have added their opinion. So I have tried deep learning and always failed. I think there are many reasons here. One important reason is that I don't have Hongtu students. I don't have Hongtu's lab. So any computationally too extensive deep learning methods, I cannot afford to do it. My students don't want to do it if it wait for like several days. And the computing center or my school will not allow me to do it. So in that sense, and another reason is like, I'm not so sure in many of those problems we have enough data because FMI and have such a, so you basically have to convert FMI to network. But my understanding that deep learning is very good for imaging, but the network fundamentally is not imaging. So I have tried, but so I will keep trying, but I just let everybody know that I was not successful. I think Jen may give some opinion how the deep learning can be very successful. Yeah.
Yeah, I don't have a lot of experience with a successful deep learning application. It just it can work. I think it's more like broadly you can consider some of like statistical method will be a special case of the deep learning. And I think you can be sort of like a broad, like you don't have to consider boundary that I'm doing statistical or doing deep learning. if you think about the linear regression, you can consider it as a special case of deep learning. You have a linear activation function, one layer, right? So that's part of the deep learning. But it's more like how you actually can using some method or some statistical tools or AI tools to solve your problem. I think it's sometimes you don't have to have a very computational intensive kind of method you don't need a very large data set. You can use some simple method to solve the problem. I think but on the other hand, of course, if you do very large data set and you need a computing resource, that's we all need it in the future. Yeah.
And in general, if you're doing computer vision and also NLP problems, I think the computer vision, the pre-trained models are very powerful. And actually, in particular, if your signal is heterogeneous, and then I think this pre-trained model is more powerful. And traditional models actually like structure the data. Usually, you assume the model, the data is very homogeneous. Otherwise, how could you run a linear model with all the signals aligned? And because that's the key differences, because you think about a language, the same sentences, you can speak in different, you talk about, John talk about another way to speak the same sentence and the meaning, right? And when you put the unstructured data inside, because for many data set, even for neuroimaging data, and you have the different people have the different, like connectivity patterns. And they have all these kinds of variations, large variation inside. Then we were early days, we use a topic models, right? Then, for NLP. Why do they introduce a topic model? Or I suggest they use finite mixture models, right? Or use basic non-parametric. It's because your data have the variations. But the topic models is another level above that. The deep learning is another level even better than topic model. That's these days in NLP, nobody use, and so few people use the topic model anymore. That's basically the key point. Okay. We just
I agree with Hong Tu. I think if you have pretty clean and homogeneous distribution the data, right? I think the statistical model probably is the best to try first, right? If it works, then you know the mechanism, very easy to understand, right? So it's explainable, right? But for some tasks, the data are very noisy and it's a mixture of a lot of different distributions, right? So like say, vision language, that kind of thing, right? So the machine learning may work, actually shows it's working, right? But sometimes the statistics can help, right? Even the machine learning, like Mu did this, right?
So the situation you just described is exactly applied to the topology data analysis. It doesn't assume any distribution assumption. It doesn't assume any model, and they extract the signal, topology signal. So
right. But I think that the thing is like a convolutional neural network is very popular, right? So it can handle a lot of ImageNet like kind of recognition. Then Mu actually you and Ant you developed this actually extended that to the manifold, right? Using Flushing Betramine or that kind of operator. I think that's probably the statistic that can help the AI machine learning. Now you can handle the data on the surface on manifold, right?
Yeah, just we have 5 minutes for conclusion remarks. I also have a survey. People, all the participants it's important that you finish the surveys for us.
Let's thank our panelists again. Thank you.
Thank you. Yeah, you want to call? Yeah. I actually, these are some concluding remarks. Yeah. Okay. You move this one here or?
I haven't used this camera before yet.
Maybe you can turn around, point to me. I think
that should work.
Okay. And thank you for all the participants. And I just want to read some of the acknowledgements. What happened? Okay. And the first one, I want to thank the Jen did most of our work. And the key reason I think is and also thanks all the organizing committee members for forming all these different invited talks sections. And that's great so far, even though we need to improve in certain degree, but overall it's a great workshop. And also thanks MSI. I think compared with last year, I think this year in terms of they improve a lot in terms of how we monitor the whole workshop day by day. Okay. Thank you. And this is really 5 days one track workshop, and actually they give people a lot of information and stress you up all the times from the early morning to late night and late afternoon. And but you see the participants here is the number of the participants are great. Okay, and we have 5 keynote speakers. I think they're really stimulating, open our eyes, and I think they've come from the different, slightly different areas, but they're all related to the human brain and everything, right? They cover a larger spectrum of the topics. All these keynote speakers are top researchers in the field. And also we have a lot of great speakers here and also senior people, okay? And also I don't cover all the speakers because I just take part of that. And hope you enjoy the pictures. Maybe we'll need to assemble them together and ask Jen to share the I will assemble all these pictures into the Google Drive and share with all the people. Okay. And also the most important part is about the panel discussions. I think we have 4 wonderful panel discussion panels and all these, and we discuss some deep questions in the field in many aspects. Okay, I hope you can learn something or you want to contribute to something. That's basically for future discussions.
What year was this picture? What year was your picture? Previous page.
Which one?
Previous page, your picture.
No, I'm not there. I forgot to do.
Keep going.
I didn't put them
The panels. The other way.
Other direction.
Panel discussion.
This one. Yeah, this picture. What year was this picture?
Was there a point?
Oh,
that's a picture I took in 2018.
Longitudinal data.
Yeah, that's longitudinal data. I need to put my picture here, right? That's aging effects. I always see it. And also, I thank all these panel members for honest, frank discussion about all the questions. It's great in particular, great for younger, basically, researchers. And I hope you really learn something and shape your future research, right? That's important. And also, everyone tried to answer these 3 questions. And I hope that you can keep on thinking how to do it, okay, for your own research. And I hope you see that when we design this workshop, we really want to cover all the whole spectrum of research. Many of the students, in particular, young researchers, not like us, like Aloja, not like people like us, I've been working in areas almost over 20 years. We can see the big pictures, and also because we interact with many top peoples, attend many different conferences, we know what's going on with the field, what are the most important questions. And I think are really important for the people, for researchers like us, even you are senior, and I need to think about what are the most important
things we need to work on, okay? And how to address all these questions, how to work with your student together, basically to tackle some of the really challenging questions. The most important thing is not you do everything. You need to do something. First thing, you need to do something more impactful. People really use your tools for solving real problems, okay? And that's the most important thing. Then at the next levels, we need to work together as the community, how to help each other and make sure that the whole community can grow. Okay, that's one of another purposes when we basically tried to have this workshop. Okay, and that's basically when I come back of a tech company. I think this is something I need to do for the community with the Chen, because I need to pass the torch to him. The next time he need the effort, I need to step back. Okay, I think this is very important for her communities basically continually to do something really impactful things for the community, for neuroscience in general. Okay, that's the most important thing. Okay, we need to think about how to do something serious and make a big impact to neuroscience and the statistical science. Okay, in what sense? Okay, everything is important from clinical trials, from design, or clinical applications, translation. All are important, and the most important things we have the time to have these social moments, and actually this is very important for our life, right? It's not just to write another papers. Actually, it's important that we gather together in the next few years, and basically have fun together, to share some of our experiences, also in terms of life and also in terms of our research. Thank you.